Hey guys, hope you're studying hard for that evolution test, because, yeah, that's all I'm going to say. So, this particular video is going to be over carbohydrates, which is one of the 4 macromolecules of life, or biomolecules as they're often referred to. Another term that you might hear would be organic molecules. They all kind of mean the same thing. So, this one's over the sweet stuff, carbs. Here we go. So what do you need to remember? The first thing you must know, carbohydrates are your body's immediate source of energy. They are energy molecules, high octane energy molecules. So all those low carb diets, yeah, about that. You don't eat a lot of carbs, or you don't eat the right kinds of carbs, you're going to find yourself losing the amount of energy that you're used to having, because that's what your body primarily breaks down to get its energy supply. In terms of what they look like, here's the generalized structure. They come in several different molecules. This particular one here is glucose. And carbs are very versatile. They can form ring structures like I've circled here for you, but they can also be linear and they can also be branched. So we'll step into that. So let's dissect the word. Carbohydrates are made of carbons, hydrogens, and oxygens, kind of like the names suggest. And it comes in a very specific formula. It is important that you know what this formula is. It says that for every carbon you will have 2 oxygens, and you will have 2 hydrogens, and you will have 1 oxygen. So for every carbon molecule you have, there will be 2 hydrogens and 1 oxygen. This is the base model, the base formula for carbohydrates. So if you think of C6H12O6, which is glucose, you can see that that's true. For every carbon molecule you have, we have 6 of them, you'll have 2 hydrogens. So 2 times 6 is 12. So that's where the 12 hydrogens come in. And then you'll have 1 oxygen. If you have 6 carbons, you're going to have 6 oxygens. So this formula always holds true for carbs. They're always found in this basic formula. What are they used for? Like I told you before, it's your body's immediate source of energy. But we're talking about fast energy. So your body breaks it down rapidly to power everything you do on a daily basis. It is also a form of energy storage, but this is going to be short-term energy storage. Your body isn't really going to hold on to carbohydrates for a prolonged period of time. They can be used for raw materials in some of our metabolic cycles that we will talk about. And they can be used as structural materials. Now you see this one way more in plants than you do in animals, and we'll talk about how they're used structurally in plants as well. The monomer, because remember I told you macromolecules are repeating chains of the same small unit over and over and over and over again. Those small units are called monomers. So the monomers for carbohydrates are sugars, and they're different types. It just depends on what you're forming. And classic examples of carbohydrates would be sugars, starches, cellulose, some other stuff. So all this should be real familiar. We're going to move on. How do you know you're working with a sugar? If the name ends in the suffix OSE, os, you are working with a sugar. So galactose, fructose, sucrose, maltose, all of those, just because of the ending alone, should tell you you are working with a sugar molecule. We name them based on how many carbons, they have. So you sometimes hear us refer to, like especially in DNA, we talk about having a pentose sugar named ribose. Well, that pentose describes how many carbons you have. So if you have 6 carbons, you're a hexose sugar. Glucose would be an example of that. If you have 5 carbons, you're a pentose sugar. Ribose would be an example of that. If you have 3 carbons, you're a triose sugar. Glyceraldehyde will be an example of that. And these are what their structures can look like. So we can have that classic hexagonal shape I always tell my kids it kind of looks like a stop sign, just to help them remember. And then we can have one with 5 sides, so it's not as obviously stop sign-ish. At least to me it's not. And then if you have 4 or less, you tend not to form a ring structure. Instead, you'll form some kind of linear structure like this. So this is what we call a ring structure. Whoops, spot ring wrong. Let me write that someplace else. These are both ring structures, and this would be linear. So we talked about functional groups in the introduction video to macromolecules, and we said that the functional group is going to change the function, obviously, of the molecule or the substance that it is attached to. So here's a good example of how that works in carbohydrates. We talked about carbonyl groups in that first macromolecule video, and I told you that the type of carbonyl depends on its location. If it's found on the end of the molecule, like it is here, and the carbonyl is specifically that C double bond O. So if we find it on the end of the molecule, and it could be either end, left or right, then it forms an aldehyde. So all of these would be aldehydes. If it's found not on the end, so if it's found in between 2 carbons, and that could be in the exact middle or anywhere where it's between 2 carbons, like it is here and here and here, instead of an aldehyde, you have formed a ketone. All right? And you can see some of the differences in terms of name. But note, regardless of what they're named, except for this guy right here, they all end, and well, some of these down here as well, they all end in that OSE. The group name for aldehyde sugars are called aldoses. Okay, so aldoses are the group name for aldehyde sugars, whereas ketoses, that's the group name for ketose sugars, or ketone, sugars. Okay, so it collectively tells you that all of these are sugar. Now we can go in and individually name them different things, like this is glyceraldehyde, which you will become familiar with when we talk about the glycolysis cycle. And this is dihydroxyacetone, which again is part of that glycolysis cycle. It's one of the things that your body does metabolically to break down sugar, but they're all classified under that group name of aldose or ketose. So, if, like I said before, if you have 5 carbons or 6 carbons in your structure, then you tend to form rings just because it's a little bit more stable. So it's a better situation to form a ring structure if you have multiple carbons, as opposed to if you only have a few and then you form a linear structure. It doesn't mean that they don't exist linearly. They do, but they're more stable in that ring form. Oops. Now, one of the things that can get confusing is to figure out which carbons we are talking about. Now, here's the other catch. When we talk about ringed structures, you have to remember that each ring structure is going to have an oxygen that is a part of the ring. It's actually in the ring. Okay? That oxygen does not get numbered. We don't count it. It's there for us primarily to be able to figure out direction. Okay, so when you have a 5-carbon ring structure, all the carbons will be in the ring. When you have a 6-carbon ring structure, one of the carbons will not be in the ring. It will sit above the ring. So that's one of the reasons why we number these. And they're numbered as primes. We always find the oxygen, and then we go down from the oxygen. So in this direction. So this carbon would be 1 prime, this one is 2 prime, 3 prime, 4 prime, 5 prime. And because this is a 6-carbon sugar, the 6th carbon sits on top of the 5th carbon, and that one would be 6 prime. Now, it's not that important for a lot of molecules, but when it comes to DNA, this arrangement of 6' here and 3' here is the whole reason that, one of the reasons I should say, that DNA fits together. So these blue lines represent bonds, which, as you know, aren't really like that in real life. Like, there's no line connecting two atoms together, but we draw them in so that we can have a visual representation of them. Now, carbon-to-carbon bonds are very, very energy-rich bonds. And when we go through cellular respiration, so not breathing, but the creation of ATP, the breaking of bonds, it's that energy that we release that we use to make ATP. So we can take our carbohydrates and break them down into two big groups, simple versus complex sugars. So simple sugars would be monosaccharides. Mono means one, and saccharide means sugar. So literally this means one sugar. And that's how many monomers a monosaccharide will have. It has just one type of monomer sugar. So a monosaccharide of glucose is just glucose repeated over and over and over and over again. Disaccharides mean two sugars. And this tells you that they have two monomers. So sucrose would be an example of a disaccharide. That would be fructose plus glucose to make sucrose. And that would be repeated over and over again. Poly, as you know, means many. So polysaccharides will have, they're pretty much just large polymers. Lots of individual monomers, maybe. Or sometimes they're just one kind of monomer. It just kind of depends on what you're making. And a good example of this would be starch. So like in potatoes or rice or pasta. We've already mentioned this, but we're going to talk about it again because it's so very important. You're going to build a carbohydrate the same way you build any macromolecule, and that is going to be through dehydration synthesis, so the removal of water. You take away that OH and that H, and you create the ability for this oxygen to bond to this empty space right here, and it'll seal the two molecules together. So dehydration synthesis builds sugars. So let's look at this example. We have glucose and glucose coming together. We're going to take that water away, create a space for that oxygen to bond, and we're going to end up with two molecules of glucose linked together, forming a disaccharide called maltose. This is important. This bond that holds these two glucose molecules together is called a glycosidic linkage. So glycosidic linkage is the bond that holds monomers together. Okay, so again, the bonds that hold monomers together is called a glycosidic linkage. Those are made through dehydration synthesis. The other term that you should hopefully remember for this is condensation reaction. All right, so this is just another example, but this time we're making sucrose. So we have two monosaccharides, glucose and fructose. We take the water away, and now we've made sucrose or table sugar. For those of you who may not have known this before, sucrose is fruit sugar. So when you eat apples or pears or oranges or whatever, that sweetness is coming from the monosaccharide fructose. Sucrose is table sugar, like what you put in coffee or tea or whatever. So a polysaccharide is a huge unit of sugar. It's a polymer of sugar. It doesn't cost a whole lot of energy to build because it's kind of like making a Lego wall. You're just linking pieces together. And it's easily reversible, which is why your body kind of uses its sugars as its quick energy, its temporary energy store, because it's easy to make them, but it's also easy to break them and release the energy that your body's going to need. Its function is mostly energy storage. Like I told you, that would be short term. And there's some very classic examples of polysaccharides in animals and plants. So in plants, you should be thinking of starch. That's our classic example of a polysaccharide. The animal version is something called glycogen, and it's found in two main locations. It's found specifically in your liver, because that's where it's made, and it's also found in your muscles. So that's in terms of energy storage function. When it comes to structure, plants, some insects, and arthropods as well, and some fungi use polysaccharides specifically for building units. So in plants, the example would be cellulose. So think of eating celery. You know how when you bite into it, there's that stringy stuff that even though you chew it up, it's still kind of really hard in your mouth and it doesn't really break down easily. That would be an example of cellulose. It's used specifically to hold that celery stalk upwards. The other kind of polysaccharide is called chitin. Now, not chitin, or some people pronounce it chitin as well, but chitin. It's found in arthropods, so things with a shell, like lobsters and shrimp and crabs, and of course our little bug friends. And then some types of fungi use it as well to form protective layers out on the outside of themselves. So that's why when you step on a bug, it makes that crunching noise because they have that exoskeleton. Well, it's the chitin in their exoskeleton that's making that crunching noise. It gives them a lot of support externally. Polysaccharides can come in two versions. There are linear polysaccharide chains and branched polysaccharide chains. So if you look up above, look at that first picture right here. This is your main chain, and then see how there's one that branches off? That would be a branched polysaccharide. And over here, there is nothing coming off of that main chain, so that would be a linear polysaccharide. This is an example of amylose and amylopectin, both of which are found in plants. If you look down at the second picture at glycogen, which is animal polysaccharides, these are highly branched. So see if you can follow me. Here's the main chain. So wherever I'm going over in red, that's the main chain. And I'm going to go over in blue where the branches are. So there's a branch here. There's one here. And here. And there. And this one connects here. And there's another one here. So their branches kind of all over. Okay. Now, the difference is in our more linear polysaccharides, these would be what we call slow release, or slow release of energy in this case. Because if you think about it, enzymes have to break these bonds that are holding all of these little molecules together. So technically, there would be a bond here, and another one there, and another one there, and there, and everywhere I have a red mark, that's where a bond would be. So you need an enzyme to come in and put the water back and break the molecule apart. Well, if I'm linear, that means an enzyme can only work from here and from this end. So I can only work with two enzymes can only work on this molecule. Maybe one can work from up here. So 3 enzymes could work on this molecule. Over here, only 2 enzymes could work on this molecule. It would take those enzymes a while to go through and break off every single little glucose molecule. So the release of energy in these molecules would be slow. Whereas if you look at the animal polysaccharide, the release here would be a lot faster because you have so many branching points. So there can be an enzyme working here and another one working there, and one working here and one working here and one working here and here and here and here, and you get the point. There's a lot of places that enzymes can start working, so they're going to be able to break and cleave off those glucose molecules at a much faster rate, which makes this faster releasing for energy. So when you think about it, plants don't require tons of energy because they don't have a whole lot of activities taking place. Whereas animals do have a lot of activities taking place. They do require faster output of energy. So it makes sense that animal polysaccharides, which is an energy store, is stored in this form versus plants, which are stored in that form. So the more branching you have, the faster digestion can take place. The faster you can break those glucose molecules off and utilize the energy that you're releasing. Now, when you first look at glucose molecules in animals versus glucose molecules in plants, or even glucose molecules in starch versus glucose molecules in sugar, they look the exact same. But on the molecular level, on the structural level, there is a very big difference. And that difference has to do with the location of their hydroxyl group on their carbon 1'. So on that first carbon, where is that OH group found? In starch, the OH group is found on the bottom of that carbon, so it's pointing down. Whereas in glucose, the OH group is found above the carbon, so it's pointing up. These two different orientations have two different names. When the OH group is pointing down, when it's on the bottom of carbon 1, it is called the alpha glucose arrangement. When the OH group is on the top of carbon 1', so it's pointing up, it is called the beta glucose arrangement. And to be very honest, this diversity in arrangement is the main difference between a starch molecule and a cellulose molecule. These guys are isomers of each other. And remember, we talked about structure determining function. Structure goes with function, and function goes with structure. So if you think about it, humans are capable of digesting starch, but we are incapable of digesting glucose, and it's because of the arrangement of that OH bond, or OH group, sorry. So when it comes to digesting starch, it's really easy for us because these starches form what we call a 1,4 alpha glucose linkage. So let's break it down. Why is it 1,4? It's 1,4 because of the arrangement of the carbon. So here's our oxygen. Remember I told you find the oxygen and then you work down from the oxygen. So this would be carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, and up here is carbon 6. Let's look at this other one. Again, we're working down from the oxygen. Carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, and carbon, whoops, sorry about that, I hit something by accident, and carbon 6. So in science, names give you a ton of information. A 1,4 alpha glucose linkage tells me that my bond is forming between my first carbon and my fourth carbon. So that's where the 1,4 linkage comes in. Alpha glucose tells me that my linkage happens underneath my carbon because those carbon molecules have OH groups that are underneath them. They are in the alpha glucose arrangement. So the enzymes that humans possess have the ability to break these 1,4 alpha linkages. That's why we can digest starch. Cellulose, on the other hand, is a whole lot harder to digest. And mammals really just don't have the enzymes that are capable of breaking those bonds. So let's look at this bottom part, and we're going to see a difference. In cellulose, we have kind of like an alternating of the linkage. In starch, it's always going to be a 1,4 alpha glucose linkage across the entire molecule. The molecule can be millions of glucoses long. Each of them will be linked by a 1,4 alpha glucose linkage. With cellulose, it's not really like that. We have a 1,4 linkage still, because we're still linking carbon 1 with carbon 4, but in some of the molecules, it's going to be a beta glucose arrangement. That bond, that glycosidic bond, is going to be formed on top of the carbon because that's where the OH groups were. The enzymes that humans possess aren't capable of breaking those beta bonds. Instead, we can break the alpha bonds. So if we had to eat cellulose, we wouldn't get a whole lot of nutritional value from it. Luckily for us, bacteria possess the enzymes necessary to break down beta-glucose linkages, and since things like ruminants, like cows and goats and sheep, and even humans, do have a steady supply of E. coli in their gut, then we are to a degree able to process some of the cellulose as food material. So cellulose is the most abundant organic compound on Earth. Every plant has cellulose in it in some form or fashion. It produces a ready food supply for our herbivores. But seeing as how most of them are mammals, and we just said that mammals don't have the ability to break these bonds that cellulose has, that creates a problem. And initially it did, but herbivores have evolved a mechanism necessary to digest cellulose. If you've ever taken a good look at a herbivorous animal, you'd realize that they have kind of like a potbelly situation going on. And it's not just because they eat tons and tons and they're lazy and they don't work out enough, but that's part of the mechanism that they've evolved. A lot of herbivores have multiple chambered stomachs, and their stomachs act like these big fermentation tanks. So they initially take in the grass or the plant material, whatever it is that they're eating, and they chew it up a little bit and they swallow it. And that greenery sits in one of their several stomachs for a prolonged period of time, where it's acted on by digestive juices and also bacteria, which we already said have the enzymes necessary to break down cellulose. So then the herbivores are going to kind of pull up a little bit of that green material after it's kind of sat there for a while. They're going to re-chew it and swallow it, and then it's going to move on to another chamber of the stomach where it gets processed some more. And this way they're able to extract as much nutritional value out of the food that they eat as possible. Carnivores, on the other hand, have not developed this ability to handle cellulose material, mostly because we hardly ever eat any. And that's one of the reasons they eat meat. They get their energy and their nutrients specifically from meat. It doesn't mean that carnivores never eat green stuff. I'm sure you've seen your dog or your cat eat green things like plants and stuff in your house or in your yard periodically. But for carnivores, it becomes a situation where it is roughage, it's fiber, in other words, as opposed to it has a direct nutritional value. Okay, so we kind of talked about this already. Cows, like I said, they're going to be one of those herbivores. Look at their stomach, big pouching out of that stomach, because really what it is just a fermentation tank. So because of these two things that they've evolved, these multiple chambered stomachs that can kind of hold food there for a prolonged period of time, and a very good mutualistic relationship with the E. coli that live in their gut, they can digest cellulose well, and they don't have the need to eat any other sugar. They get all their nutritional value from the grass and the plants that they eat. Gorillas, on the other hand, are omnivores like we are. So while they can digest cellulose a little bit, they definitely can't digest it well enough to get a ton of nutritional value from it. And thus, they must add another sugar source like fruit to their diet. So that's how they get their immediate energy. Okay, so we talked about this. A lot of herbivores have bacteria that naturally live in their digestive systems, and they help to digest the meals that they eat in the form of grass or plants. Let's talk about this coprophage really quick. Some animals, like bunny rabbits, for example, they don't have as many stomachs, and they can't really sit around and wait for their food to just naturally digest on their own. So they run it through their system, they get rid of it, and then they eat it again and run it through one more time. So yes, they eat their own poop, but that way they really get a chance to pull as much nutrients out of it as possible. All right, hopefully this was fairly straightforward and easy. Don't forget you have a test on Monday and Tuesday. Hopefully that's what you're studying for, and you're listening to this video on either Monday night or Tuesday night after you're done with your test, because we're not going to talk about it until Wednesday or Thursday. So see you in class. Bye.